Okay, for this section of the class, we're going to take a look at how we can uh, look at how the immune system might be suppressed, or how we can take advantage of it to stop the reactions that we've wanted it to take. In particular, we've wanted it to take action against pathogens or cancer cells. And now what we want to do is look at a part of medicine that deals with the suppression of the immune response. And there's basically four take-home messages for this. The first are that MHCs, which have another name called HLAs, are key determinants of organ donor acceptability. Two, effective transplant therapy depends on understanding the reasons for immune rejection and, if necessary, suppressing this reaction. And three, natural immunosuppression can result from genetic defects affecting the function of the immune cells. Pathogens can induce, and fourth, pathogens can induce immunosuppression. And so we're going to take a look at these, and so these are the things you want to keep in mind as we go through this section. Now, these are two interesting, these Dylan and Dawson. There are a couple of twins, cute twins here, and they have an interesting connection between Dylan Dawson and cheetahs, and that has to do with their immune systems, and it's something that you might see that's common for twins, and unfortunately, it's also now has been found to be somewhat common between cheetahs too. The first is that when you take, in the early days immunology, it was found that you could perform skin grafts from one to the other with no tissue rejection, so that in fact, it was kind of a novelty in medicine that to do any sort of transplantations that you needed to have in the early days a twin. So you could take from two genetically identical animals or mice, or in this case two genetically identical twins, and it was found you could transplant skin from one to the other and you wouldn't get a rejection response. What's been interesting in that, however, for many years meant that, of course, that you couldn't do anything you couldn't use transplantation at all, because obviously you needed some way to suppress the immune response or understand it. And until that was done, it was really a backwater of medical science. Now, in the 1990s, early 1990s or so, mid-1990s, it was found that cheetahs also are very, very genetically similar. And this was found when in the region where cheetahs are their main territory in Africa, that game wardens, they had a reason to potentially do a skin graft on one cheetah onto another one, and because of injuries or that the animals had suffered, of course they didn't know, in fact, that they didn't know that they had any genetic similarity. And what they found is that in fact these skin grafts took. As they got into more investigation, they found that in fact the genetics between the cheetahs is very, very limited. Genetic differences is very limited. In fact, they're so similar that you can do actually transplant from one to the other, and they don't have to be necessarily in the same family. This, of course, is kind of catastrophic for the cheetah, because it means its diversity in its immune response is quite low. Now, what's been reasoned is that there must have occurred at some point in the cheetahs millennia ago some event, maybe a viral infection, that wiped out all cheetahs except for a few remaining ones that were highly genetically similar. And it was from that population that present-day cheetahs are as we know them. What that means, though, is that cheetahs are as well as twins, are in a black box, in sort of in a box, because they can't. The genetic diversity between them means their immune response between them is very similar, and therefore, if there are holes, for example, there are antibodies that they don't necessarily have the right antibody diversity. They don't have the T cell receptor diversity, MHC diversity. Then that leaves them susceptible and open to a potential viral infection that could wipe them out easily, because there's not that much. And so that's why there's been concern about that with these animals, because they boxed themselves in genetically. They survived, and surviving the infection before, they boxed themselves in genetically. So it's actually made them more susceptible to potentially extinction due to a viral infection. Now, what is this suppression? What is this immunosuppression? What is this when you have an immune reaction, when you have rejection? What does that mean? And we're going to find out. Well, first, let's take a look at how we identify and look at graphs when we're looking at graphs between individuals. And that nature of that graph means that it's evident. There's evidence, basically, from what I described, is that if two individuals are genetically diverse, then you can't, for example, do a skin graft. Now, the evidence that graft rejection is immunological comes from a couple of things. One is that the second rejection, if you do a first transplantation, you get rejection, but the second rejection, actually, if you tried to do it again, is faster on second exposure. And you know why this is the case, because it's a secondary response, so it's quicker and faster. And we also know that agents that suppress immunity, suppress the immune response, prolong graft survival. So those are the two things. These are two reasons that are consistent with the fact that graft rejection is immunological. And third, tissue grafts being rejected are, if you look at them, are usually invaded or just chock full of immune active cells that have invaded those tissues, trying to destroy them. So if we look at the types of grafts, one type of graft is called an autograft. This is a graft onto a donor. So this would be, for example, a skin for a burn victim. So this is graft from yourself onto another. So if you got burned on the arm, for example, you could take skin from the leg and transplant it in, and replace that skin on your arm with the skin on your leg. So that's referred to as an autograft. So that's a graft onto a donor. Second type are called isografts, and they're between syngenetic individuals, closely genetically similar individuals. And so these are, these are grafts in which the person is genetically very similar, and these might be from a mother to a parent to a son or daughter, or it may be a daughter or son. It's between siblings, between a brother and a sister, or very, very close, potentially cousin. And these are typically what typical examples of these would be a kidney transplant or a bone marrow transplant. In some cases, you see this. You've seen this where people have actually had children in anticipation that the child might be genetically similar enough to the other child in order to do a bone marrow transplant. Also, I might add, isografts, liver has become more prominent as an isograft, because you can take part of the liver and it can regenerate. You don't have to take all of a person's liver, and you can give that to a person, transplant to a person, and it will start to function as the patient's liver action. The other one are allografts, and these are from the same species. So these would be, for example, heart or liver. And so these would be ones in which heart, lungs, and potentially a liver, in which in the case of the patient, the donor generally is not of the same, is not genetically similar, but is able to give a functioning heart or liver or lungs to the patient. In that case, they have to be under fairly extensive immunosuppression therapy in order for that to actually work. We're going to talk about that. But those are referred to as allografts. The last category are xenografts. These are from different species. So these would be ones, for example, pig heart valves, and potentially in the future the use of genetically engineered animals, most likely pigs, because their organs tend to be about similar in size to humans, and nobody really cares that much about pigs. If it was transplants between primates, it would be much more of an uproar over that. But for pigs, there has been a lot of interest in that, in genetically engineering them so that they could provide like pancreas or liver or heart, at least on a temporary basis, to potential patients. So autografts, isografts, allografts, and xenografts. Now, the most important sets of antigens for tissue rejection, as it turns out, are the complexes you already know about. Those are the MHC1 and MHC2. That's one of the reasons we spent some time talking about those. They're also known as the human leukocyte antigens, or HLA. So you'll see HLA testing. That's the same thing as MH1 or MHC1 and MHC2. So MHC1 equals HLA. So whenever you see that, that's what and there are different categories of HLA, and that's what we're gonna look in as we go on here. Now, the different categories of HLA are grouped into class 1, class 2, and class 3. Class 1 are the MHC1s, or class HLA1, and there are A, B, and C types of those. For the class 2, or HLA2, these are HLA2 DR, DQ, and DP. So, and then there's class 3, which is really complement factors, and these are not important. They're not involved in graft rejection. So these two up here are the ones that you really want to pay attention to. One easy way to remember this, if you forget, which one's HLA1, which one's HLA2, or MHC1, MHC2, is that if it has two letters, it's 2. It's class 2. So if it has two letters here, it's class 2. And you'll see there's no D in class 1. So these are the ones we're going to concentrate on here, class 1 and class 2. Now let's remember what MHC1 class 1 is. It's composed of a one polypeptide chain, which has an alpha 1, alpha 2, and alpha 3 domains. It also has this beta 2 microglobulin, and this all sticks into the membrane, through the membrane, and has a cytoplasmic tail. And again, the peptide binding region is between these two alpha helical regions and this beta sheet region. And again, you can see. I thought it was going to move, but again, you can see it's all one polypeptide, and held in this microglobulin, keeps this kind of stabilizes this structure with the peptide binding up here. Now, if we look at genetically this, there are actually 3 types of these. Depending on the individual, there's basically 3 types, A, B, and C. And so you can produce, as it turns out, produce A, B, and C. So the HLA-A, HLA-B, HLA-C. And so these are going to be three that have unique and different abilities to bind to peptides. Now, everybody, I'm sure, is diploid, and so you have a mother and you have a father. So if we look then, because you're diploid, you're going to produce A, B, and C from the mother and A, B, and C recombinants from the father. So therefore, you will produce basically a maternal HLA A, B, and C, and you'll produce a paternal-based HLA B and C, and of course the recombinations of these they happen. So in large part that means you'll get six. You'll get six different HLAs that can be expressed, and these can be expressed, of course, by all nucleated cells, and so all nucleated cells can express the HLA A, B, and C, which are MHC1. So what does that mean? So if you had a virus that's been the cells are infected with and the peptides are produced, there is at least six different MHC1s that can sample that peptide and potentially display it depending on their specificities. Now, there's a great deal of polymorphism in these genes, and this is where, and so it's important to understand which one of these genes has the most polymorphism, has the most differences in population. And it turns out that they're all quite a number of polymorphisms in there. And this is just this is a snapshot at one time. But you can see A and B have the most polymorphism. So if I were to look into a population, just say in class, I would expect to see more differences in the B among individuals, followed by A, followed by potentially C. And so that's where within the population this great deal of this polymorphism allows the population to have a diversity, so that although some people might die, the population will be protected by the diversity. And so the species will be protected from death, from being wiped out, because there's diversity within that population. And that's why even with Ebola, for example, you saw some people actually recovered. Some people, it's a contributing factor that some were able to fight, their immune systems were able to fight off, while others quickly succumbed. But it wasn't it's not 100% fatal. So that variability that we're talking about, this variability that you see in here, this is actually, as not surprisingly, that variability is in the binding pocket, in this region here, the binding pocket for the peptide. There's variability along the beta sheet floor, and there's variability along the alpha helices. So those are the regions where you see the most diversity, and therefore, and that's what you would expect, because that's the regions that bind to the peptide. Now, if we look at class 2 is composed of 2 polypeptides, and each has 2 domains apiece, and the polypeptides have to come together to form the class 2 complex. And again, you can see here the red, there's 2 chains, the red and the blue, that make up the binding pocket for the peptide. And so you need contributions, basically 50% from one and 50% of the other. So if we look at these genes come across as DP, DQ, and DR, and that means that, you remember, there are 2 chains, they're not exactly the same. So there's 2 chains that are alpha and beta. Just remember, there is an alpha and beta chain. There's DQ has an alpha and beta chain, and DR has the most diversity because it has an alpha, beta, and a beta prime. And it can have others. There can be other betas, beta 2, 3, and 4, that are that are able to form with the alpha, with the DR alpha, multiple different DRs. So as a consequence, like for the DP, you could form, there's one, or DQ, you can form one, and for the DR, you can form at least, you can form two here, right? Now, the thing about this is that you do have there is diversity among these, because the alpha chain sometimes there's promiscuity because they're not one polypeptide. You can have, in some cases, for example, for these betas, you can have different betas. You can have betas potentially binding with other alphas. And so it's not entirely strictly forbidden, but there is some basically promiscuity between the alphas and betas getting together. And this can create basically even more diversity within the expressing antigen processing cell. So if we take that one step further and look at the mother and the father, now combining this as a diploid, you then could express minimally, could express 8 of these. There could be 8 of these, but there's in fact many more. And you can have the father's alpha now, for example, binding to the mother's beta, and vice versa. And so that can double the amount for each one of those. And so in practice, because of this promiscuity and the beta chains and the number of beta chains, and the number of beta, the higher degree of the number of beta chains, diversity for MHC2s can range anywhere from 8 to 16 to 20. And it's usually somewhere up there, in the 20 or so, 20 plus. And so what you find is for the MHC2s, for the antigen processing cells, that they can, they have a great range of different peptides that they can take up and then present to T cells, right? And so if they're expressing this, what that means is that if a virus, say a virus infects a cell, then it produces several, say 3 different, say it produces 3, there are 3 for one protein, there's 3 antigenic peptides that can bind to MHC2s. When they do so, they bind to their paired MHC2, and then all 3 could be displayed depending on how much of that antigen is there. And all 3 could be displayed on the surface. So antigen processing cells have that ability to really put up a lot of different, to scan and put up a lot of different these peptides. It's true for regular cells too, for MHC1, but it's less. There's less diversity there. There's less numbers. There's just less number of potential MHCs there that can do it. But you certainly could have an MHC1 A and B, for example, could bind to 2 different peptides, and they could be displayed. In each case, that display means that they have improved their chances of, when they run into a T cell receptor runs into them, of reacting with that T cell receptor and reacting with that T cell receptor and then enhancing the immune response. So the polymorphisms for the HLA-2s are in these beta chains primarily, and in particular the DRB1. See here, there's a DRB3, DRB4, DRB5. There's a whole bunch of them. This DRB1 is where you see a lot of diversity there, and on the B or the DP, you see it in the beta 1. So there's a lot more. There's a lot of diversity. A lot more diversity in the betas than there are in the alphas. Not clear why that's the case, but you can see that's clearly there's diversity across the board and a lot of diversity in the betas. So of course you can have this can create a tremendous diversity of different MHCs, 2s, that can be displayed on the antigen processing cells that we talked about. So MHC1 has 6 variations expressed on all nucleated cells, at least 6. MHC2 has many more, from 8 to 20 or more variations expressed on all APCs. and certain cells that have been induced to produce those. We've sort of seen those in the thymus, those stromal cells, epithelial cells that can produce MHC2 on their surface under sort of strict conditions in the thymus. But other than that, it's just the antigen processing cells. Because they're most polymorphic, the most important HLAs for transplant are HLA A, B, C, and DR. So if you're looking for a match, what you would like to have is a match of those. Those would be the ones that you'd really like to have a good match on. Now you can do HLA testing, so I can take some blood from you and me, and that allows us to do some HLA testing to see how well we would potentially be a donor for someone. So class 1 antigens can be detected serologically, antibodies. They are identified by immunofluorescence or other antibody tests. For class 2 antigens are detected by the MLR response. And what is the MLR? Well, the MLR is a multiple lymphocyte reaction, and there's two different types of that. The first one is two-way MLR, and so if we were to look at this, if we took a recipient lymphocyte and lymphocytes, and the recipient would have B and T cells. The donor lymphocytes would have T cells and B cells as well. And if we mix those, allow them to interact with one another, then add in a radio-labeled thymidine. Thymidine, of course, is a nucleoside that if DNA synthesis happens, if you get proliferation, will be taken up and then incorporated into the growing DNA and the DNA because you have increased DNA synthesis because of different you get cell proliferation, and so you can measure the increase in the amount of thymidine uptake, and that'll give you an idea of what has happened. So if there's proliferation and they recognize each other as foreign, so if I mix two cells from two individuals, you'll see the T cells will interact with the B cells from the other one and vice versa. And as a consequence, they'll recognize those MHC2s. And the proliferation within means that they essentially, if they recognize them, that they are foreign, and therefore it's not going to be a good donor. If, however, you see no proliferation, and you see no proliferation of those cells, and no activation of the immune response from those cells, immune activation, then no proliferation, then they're recognized as being a reasonable donor, or it's a mild response, then they're potentially a donor. There's one other way to do this. It's called one-way MLR, and that involves when you take the donor cells, lymphocytes, and you treat those with a toxic drug, a mitomycin. In this case, mitomycin is an anti-cancer drug, and what it does is it inserts itself into the DNA, the chromosome, the DNA, and basically locks the DNA such that the DNA can't come apart, and so the cells can't divide, and essentially they will die. But if you take the recipient and you take the donor cells, which have been treated, there are cells that can't proliferate. And if you again add the nucleoside, radio-labeled nucleoside, thymidine, you can measure by looking at the amount of incorporation, amount of proliferation, non-proliferation. So if you see lots of the nucleoside radio-label incorporated into the cells, that means proliferation has occurred, and so the recipient lymphocytes therefore are not a donor. These cannot be the donor and the recipient will reject each other. And this, when you do it this way, the cells that you monitor are now strictly from the recipient, so you know what the response is going to be from the recipient. For if you see no proliferation, of course, of either, then you know that the donor is like these lymphocytes are like the person is likely to be a donor. And so even though this is done as a blood reaction, it's true for any of the organs. So, for example, it's true, that you won't. It tells you whether the person in general would be a good donor for that one. It could be heart, it could be liver, it could be whatever you want. It's not just about the blood, but it's a pretty easy test to do. Now, so we're going to stop here. We're going to stop here, and why don't you review, take these review questions, and then answer them. Stop and take it, answer them. And I'm going to stop this part. I'll finish up here. I'll show the answers, and this will end the first segment on immunosuppression.